Today we'll discuss about the development of the male and female gametophyte in Cycas. In Cycas, gametophytic development is a very important part of its life cycle. It is characterized by a distinct male and female gametophyte. Cycas exhibits heterospory, that is, it produces two different types of spores, microspores and megaspores. The microspores develops into the male gametophyte and megaspores develops into the female gametophyte. So microspore is the first cell of the male gametophyte and we can say that megaspore is the first cell of the female gametophyte. Now let us study the development of a male gametophyte. The microspore or the spore produced from the male corn, it divides into two unequal cells. There is a small prothallal cell and a large antheridial cell. So the microspore produced from the cycas, it divides into two unequal cells, a small cell known as prothallal cell and a large cell called as the antheridial cell. The antheridial cell again divides to form a small generative cell and a large tube cell. Okay. So there are generative cell, tube cell, and the generative and tube cells are produced from the antheridial cell, whereas the prothallal cell remains like that. So the microspore becomes the three-celled. Okay. So it is in this three-cell stage that the microspores are liberated from the microsporangium seen in the male corn. Further development of the microspore or the three-celled microspore develops in the pollen chamber of the ovule, and this happens after fertilization. We know that the pollination in Cycas is anemophilous. So what is anemophilous pollination? Pollination assisted with the help of wind. So at the time of the pollination, the nucellar beak seen at the tip of the ovule, it produces a drop of mucilage. And this mucilage or this watery fluid, it oozes out from the micropyle in the form of a pollination drop. And the microspores which are carried by the air gets entangled in the pollination drop. So the three-cell microspore starts its germination on the nucellar surface of the ovule. So first the microspore, it is a haploid cell. It divides into two unequal cells known as a small prothallal cell and a large antheridial cell. The prothallal cell remains like that, whereas the antheridial cell divides to form two generative cell and a large tube cell. So there is a small generative cell and a large tube cell. So the microspores are thus liberated at the three-celled condition. So once the microspore, the three-cell microspore reaches the micropylar region, the exane ruptures and intane comes out in the form of a pollen tube. So we know that the microspore, in the previous classes we know that the microspore consists of two wall layers, exane and intine. So once the microspore, the three-cell microspore reaches the micropylar region of the ovule, the exane breaks down and the intane comes out or protrudes in the form of a pollen tube. Now what happens to the generative cell? The generative cell divides to form a stalk cell and a body cell. So just before fertilization, this body cell, it again divides to form two male gametes or anthrozoids. The anthrozoids or male gametes of Cycas are naked structures, top shaped with many cilia, and they are haploid also. So once the microspore reaches the micropylar region, the exane breaks and intane comes out in the form of a pollen tube, and the generative cell divides to form a stalk cell and a body cell. The body cell now divides to form two anthrozoids. Thus, the fully developed male gametophyte of Cycas consists of a small prothallal cell, stalk cell, a tube nucleus, and two male gametes. The male gametes of Cycas are the largest male gamete in the plant kingdom, and these male gametes have cilia all around them. So it is a very important feature of the Cycas, and it is the presence of cilia that helps the male gamete to move within the ovule. So the Cycas male gametophyte is a highly reduced structure. Now we'll discuss about the development of the female gametophyte. In Cycas, the female gametophyte develops from a haploid functional megaspore within the ovule. So the megaspore is the first cell of the female gametophyte. So what is the megaspore? Megaspore is a haploid cell produced from meiosis of the megaspore mother cell within the ovule. We discussed about this in the last class, about the formation of megaspore in the previous classes. So the female gametophyte's journey, it begins with a megaspore. Megaspore is a haploid cell produced from meiosis within the ovule. Now what happens to this functional megaspore? This megaspore, the nucleus or the haploid nucleus of megaspore undergoes free nuclear divisions to form a large number of nuclei. So a multinucleate structure is formed. Initially it is without any wall formation. Okay, so you can find a large number of free nuclei. Now what happens is wall formation happen around the nuclei. So in the meantime, a vacuole also develops in the central part of the megaspore, and the wall formation starts from periphery towards the center. So as a result, the multinucleate structure will form a multicellular tissue known as a female gametophyte or endosperm. Now in the female gametophyte, at the micropilar region, female reproductive structure known as archegonia are developed. The mature archegonium consists of a venter and a neck. The neck is formed of two neck cells, and the venter has a venter canal nucleus and an egg. The egg is said to be haploid. So within the endosperm, the female sex organs or archegonia are formed, and it is this archegonia that contains the egg cell. Thus, the megaspore develops into a multicellular female gametophyte within the megasporangium, and the megasporangium or ovules of Cycas are said to be the largest ovules in the plant kingdom. So, this is about the development of the female gametophyte. Now, coming to the fertilization of Cycas. The pollen tube with two sperms and tube nuclei grow downward, penetrating the tissue of the pollen chamber. So at the time of fertilization, the pollen tube carries the two sperms and it penetrates the tissue of the pollen chamber. At the tip, the pollen tube discharges, it breaks and discharges its content into the archegonial chamber. So it liberates two sperms, and one of the sperms fuses with the egg. So one anthrozoid fuses with the egg to produce a diploid zygote. The fertilized egg or zygote is thus the first cell of the sporophyte. You know that in Cycas, the sporophytic development or the sporophyte is said to be the dominant phase of the life cycle, and the zygote is the starting cell or first cell of the sporophytic development. This zygote divides and it produces a mass of embryonal cell called as the proembryo. This proembryo has three regions. The upper region is called the hostorial region. The purpose of or the function of hostorial region is to absorb food for the developing embryo. The middle region is called the suspensor region, and it forms the long coiled suspensor. It is this suspensor that pushes the embryo into the endosperm. And then there is an inner embryonal region. This embryonal region contains meristematic cells which later forms the embryo. So the embryo consists of radicle, plumule and cotyledons. After the fertilization, the ovule is thus converted into seed. The embryo thus consists of a radicle, plumule and cotyledon. So after fertilization, the ovule is converted into seed, and the integuments of the ovule later forms the seed coat. So what is actually seed coat of a seed? Seed coat is actually the integuments of the fertilized ovule, and the nucellus is used up during the embryo development. So we can't find any nucellus, and inside the seed endosperm is present. And within the endosperm you can find the embryo. So it is the seed that develops into the young sporophytic plant. This the structure of the Cycas seed is, it's a very simple structure. There is a protective outer layer which is termed as the seed coat or tegmen, and then you can find an inner layer that is termed as a sarcotesta, and beneath the sarcotesta there is an endosperm. Endosperm is the stored food region, and inside the beneath the endosperm there is the embryo. So the embryo lies embedded in the endosperm, and it is this embryo that develops into the future sporophyte or a new plant, and the seed is albuminous. That is, here the food is stored in the endosperm and not in the cotyledons, and you can see that there are two cotyledons. So cycas seeds are said to be dicotyledonous seeds. The radicle of the immature embryo will develop into the embryonic root, and the cotyledons will develop into the seed leaves, and plumule they will function as the embryonic shoot. So this cycas seed will ultimately develops into a young sporophytic plant. So in cycas, it is a sporophytic plant, that is the predominant phase is a sporophytic phase. It is said to be diploid, and the sporophyte regularly alternates with the gametophyte to complete the life cycle. So you can find a regular alternation of generation in cycas, and the sporophyte is dioecious, that is the male and female plants are said to be separate. The male plant bears the male cones, and from the male cone there are the microsporangium containing the microspores and microspore mother cell. This microspore mothers will undergo meiosis to form the microspores, which are haploid. Similarly, the female plant produces megasporophylls. The megasporophylls have megasporangia. The megasporangia produce megaspore mother cell, which undergo meiosis to form megaspores, and the megaspores are haploid. Now, the microspore will develop into male gametophyte, and the megaspore will develop into the female gametophyte. The gametophytes reproduce sexually to produce male and female gametes termed spermatozoids and eggs. They are haploid. The sperm fuses with the egg to produce the zygote. The zygote is said to be diploid. It produces the embryo, and the embryo will grow into the diploid sporophyte. So this is the life cycle of the cycas. Now coming to the economic importance of cycas. We know that many species of cycas plants are grown as ornamental plants in many parts of the world. Cycas revoluta is a very good example for that, and cycas is also used as a source of food in many countries. There is a starch which is extracted from the stem of cycas, and it is termed as sago. In Japan, the seeds and stems of Cycas revoluta are used for preparing wine, and the juice obtained from the young leaves of Cycas urcinalis is used in skin diseases to cure the blood vomiting, and in stomach disorders also this juice is used to cure these diseases. Thus, in cycas, there is clear alternation of generation. The plant is said to be sporophytic, it is asexual and diploid. The sporophytic plant regularly alternate with the gametophyte to complete the life cycle. So, we can say that in cycas, there is regular alternation of generation. Thus, in the previous classes, we discussed in detail about the cycas morphology, anatomy, and reproduction. So, what are the primitive features of cycas? Cycas have unbranched stem with thick, persistent leaf bases, and you can also find circinate vernation of leaflets. So, this is also a primitive character. Then, presence of meso bundles in the leaf, and you can find microsporangia aggregated to form sori. Okay, so these are some of the primitive features of cycas, and of course there is a presence of motile and ciliated sperms. The anthrozoids are said to be motile and ciliated. So all these are the primitive features of cycas. So this is about the life cycle of cycas. Thank you so much.